Steroid hormones, as we've seen examples before, this ability for them to um, save neurons from cellular death. And also further, they can also support the growth, survival, and differentiation of developing and mature neurons. This ability is called, because of this ability, we label them as neurotrophic factors. They have neurotrophic effects. Neurotrophic effects is just that, supporting the growth, survival, and differentiation of neurons. Which is no surprise. We haven't put a name to it yet, but we've seen it, right? Like in the European robin, testosterone given to females, and they have larger nuclei in the brain that are associated with song learning. Right, just over the course of less than 2 weeks, the testosterone has increased the size of these areas of the brain. So they're somehow promoting the growth and differentiation of these neurons. In general, estradiol is a huge neurotrophic factor in the nervous system, especially during early development. Here we can see what they call hypothalamic explants. So these are nervous tissue that was taken from a hypothalamus and put into a petri dish on a growth medium, so that the cells could continue to live and metabolize. And if you add estradiol to the growth medium, the neurons dramatically increase their size, their arborization, and their number. And we see this plastically. Plastic, I'm not sure that's a name, but we see this ability in the plasticity in the brain. In a well-studied example of this is the effect of natural endogenous estradiol and its effects in the hippocampus. And female mammals that are undergoing estrus. This right here is an example in rodents, in rat hippocampus. And so female rats go through an estrus cycle. This is their cycle associated with ovulation, reproduction. And what we see is that the synaptic connections of neurons in the hippocampus increase in the female rat hippocampus with the increases of estradiol concentrations during their cycle. You can see the natural wax and wane of estradiol during the estrus cycle, where it reaches a peak during proestrus, so right before ovulation, or it peaks right as ovulation occurs in this transition between proestrus and estrus. And we can see, if you take a look at the close-up of one of our axons of our hippocampal neuron, A and C have a relatively fewer abundance of these axon terminals, these projections off of the axon, and these would be synapsing with other neurons. These little neuronal or axonal buttons or axonal terminals. So much fewer of them during A and C, which corresponds to our cycle, diestrus and estrus, which are relatively low points during the natural estrus cycle, and the concentrations of estradiol. But during proestrus B, which is our middle close-up of the axon, we see a very stark, noticeable increase in the concentration of axon terminals, corresponding to the peak of estradiol concentrations. So this difference waxes and wanes over the course of the typical estrus cycle in rodents. Now you might ask yourself, what is the functional significance of this? Why would a female rodent want to have this correlation between the arborization of their hippocampal neurons during the estrus cycle? What's the functional utility of this? If you want, feel free to pause and think about it yourself and see if you can come up with an answer. And I'll, before I tell you. And so, the hippocampus is a part of the brain that's important in memory. And it's very, very important for spatial memory. So remembering where things are, even if you're not actively around in those areas. It helps organisms remember where events occurred and where to go back, maybe to find certain things. As an example, in animals that cache lots of food, such as our black-capped chickadee, which caches tons of food so it can survive the winter. Same with blue jays. They have relatively larger hippocampuses, or hippocampi. With the rodent, female rodent, during proestrus, they are the most receptive to males. Males mark their territories with urine across their territory, and females smell this. They smell the pheromones that are left in the urine. And what they do is leading up to sort of finding a male, they likely come across many different males and come across their distinct pheromones. And females go through female mate choice, which is very common throughout the animal kingdom, where females are the choosier sex. They typically have the opportunity to choose from multiple males. And so the female rodent likely is going to choose her preferred male based on the smell that she likes the most. And she needs to remember where these smells were as she's traveled around to different territories. She needs to remember where her favorite smell was so she can go back there and look for and signal the male. And so it's thought that this association between high estradiol increasing the arborization of the hippocampus is going to increase the female's spatial memory ability, which will facilitate her mate choice in finding the male that she has chosen during a period where she is most receptive to the male and the most fertile. So here's a question that I'd like you to pause and think about. Why doesn't estradiol from the maternal ovaries induce male typical phenotypic development? Something in the field that they've called for a long time masculinization. And we can have a whole discussion about the problems of this terminology. But that aside, this is still something that you see in the literature, and so you should know what it means. And so the masculinization just means the development of any kind of anatomy or structure that more closely mirrors a typical male trajectory than a female trajectory. And so what we see is that, as we've shown before, if you inject females with testosterone, especially during early development, they develop this neural architecture that more closely resembles typical males than typical females. We've also learned that it's not actually the testosterone that's doing this. All that testosterone gets converted to estradiol. So it's the estradiol that's causing these structures to look more like a female, that are causing these structures to look more like a male typical structure in a female. And so if estradiol is doing this, then why is it that in mammals, estradiol from the maternal ovaries are not doing the same thing? All mammals in utero are sharing a blood supply with the female. Among the things that are circulating in the bloodstream from those maternal ovaries is high levels of estradiol. So if maternal estradiol is circulating in the female, and because of the shared blood supply, are also circulating in the developing fetus, why don't we see female fetuses experience the same development of male typical neurostructures? I mean, we don't. But I'm asking you what do you think's going on here? It seems like everything that we've discussed so far, that should happen. Estradiol should, in quotes, masculinize any developing female embryos within the womb of the female. But they don't. Why do you think that is? So feel free to pause here to think about that. Then I'm going to move on and discuss what it does. So now that you've had some time to think about it, let's take a look at the mechanism that prevents this from happening. This has been well studied in rodent models, and there is a protein called alpha-fetoprotein in rats and mice, AFP. AFP binds to estradiol. It renders it inactive, but it does not bind to testosterone. It prevents estradiol. When estradiol is bound to AFP, this complex cannot pass the blood-brain barrier, so it can't enter the nervous system. Even if it could, though, estradiol is bound up and is unable to connect with estrogen receptors. So it would still be inactive even if it could cross the blood-brain barrier. AFP is produced by the placenta and the fetal hepatocytes in great amounts. Hepatocytes are the functional cellular units of the liver. And so this increases in concentration, and before estradiol from the maternal ovaries can even think about getting into the nervous system of the developing fetus, it meets this AFP and gets bound up, right? But it doesn't affect T. There isn't much testosterone floating around in the maternal circulation. And there isn't much testosterone floating around in circulation in the developing fetus. But there is in the developing male fetus, whereas it is not in the female fetus. And that's because in the male fetus, there are developing testes. Those testes are functional, and they are creating testosterone. We can create knockout mice, mutants that are unable to make AFP. Females that are unable to make AFP, they do not show lordosis as adults, which is what you would expect if a female developing embryo were exposed to high levels of estradiol in the brain. It would change the neuroanatomy, the neural architecture, and it would have an organizational effect that would prevent the mechanism that is involved in lordosis. AFP mutant mice, females, whose mothers were given aromatase inhibitor during pregnancy exhibit normal female sexual behavior. So what does this mean? Well, like we discussed before, when these mutant mice, they don't have the protective effects of AFP, so the maternal estradiol enters the nervous system and can change the trajectory of the neural architecture in a male typical way. However, the maternal ovaries are making estradiol. They synthesize estradiol from cholesterol, which I mentioned before. To get to estradiol, you necessarily have to go through testosterone. So in the ovaries, it's a multi-step process from cholesterol to estradiol. The last step is you've synthesized testosterone from dihydrogen testosterone, and then from testosterone to estradiol from aromatase. So there's aromatase in the ovaries. You need aromatase if you want estradiol. If you give aromatase inhibitor, something that blocks this enzyme, then there's no way for the ovaries to make estradiol. So in these AFP knockout mutants, they are, through this aromatase inhibitor, are unable to make estradiol. So without estradiol, the developing embryos, the developing fetuses in the AFP knockout females never see estradiol. So they never experience the developmental effects, the change in their neural trajectory due to estradiol. And as a result, they don't experience that as they would term it, masculinization. They don't experience that. And they develop what would be considered a typical female neural architecture, and as a result, experience the typical female sexual behavior, specifically lordosis. In humans, the analog is the steroid hormone binding globulin that we find in primates and in humans, including humans. Humans are primates as well. And it serves the same function, as very similar structurally. And so we talked about in some length about the sexual dimorphic region and the song nuclei that are important for song learning and song production and avian passerines. And so I just wanted here at the end to give one example, just to make clear that dimorphic brain regions are not always larger in males compared to females. And one example of this in mammals is the anterior ventral paraventricular nucleus, the AVPV. This is a part of the brain that's involved in ovulation, regulation of reproduction, and is much larger in females than in males. As you can see in this brain section of a mouse on the left, AVPV are the dark stained cell bodies, much more numerous and concentrated than what we see in the typical male section. The AVPV developmentally is the female could be considered sort of the default because it's high levels of testosterone exposure, which induces cellular death or apoptosis, causing the reduction in the size of the AVPV in males. Now this wraps up the end of this recorded part of our lecture. I had intended today to discuss or lecture quite a bit on the board about the hypothalamic pituitary axis, which is kind of the endocrine sort of like master center of the body. So I'll be working on that. I think I'll be able to record myself drawing it, and then posting that lecture as well. But hopefully I'll be able to get this exported and up in time for you to actually watch it during the normal class hours. All right, so thank you again, and sorry for the inconvenience. I know this isn't quite as entertaining or enthralling, listening to my sick voice on YouTube. But I'll get that other portion of the lecture up soon.